Thank you again for taking the time today to join us for another NanoTemper eDemo. NanoTemper's technology is all about helping researchers characterize in-solution stability and affinities. And today we are going to explore how our Prometheus Panta and Monolith instruments work harmoniously together to create an ecosystem that allows researchers to flourish and more quickly and efficiently do good science. Ultimately, our time is precious, and we know that time is money. So by troubleshooting and assessing the stability and functionality of candidate molecules throughout the entire biologics, structural biology, or drug development processes, we can accelerate the time frame from identifying therapeutic targets to getting them to the market, reducing the cost significantly from early development and lead discovery all the way to manufacturing and clinical release. And by using the Prometheus Panta to confirm preliminary hits and assess the thermal stability, aggregation propensity, and oligomerization of proteins, researchers can confidently move on to acquiring binding affinities with the monolith, effectively de-risking these affinity assays. How do the instruments work? We're going to start with the Prometheus Panta, which uses 3 different optical techniques to probe conformational colloidal stability, and they are nano-DSF, back reflection, and DLS. So nano-DSF, or differential scanning fluorimetry, is used to detect subtle changes in the fluorescence of tryptophan and tyrosine residues present in virtually all proteins. The fluorescence of tryptophan is strongly dependent on the protein solvent environment, and by following changes in the fluorescence at 330 and 350 nanometers, representative of the folded and the unfolded states respectively, thermal stability can be assessed label-free. So this dual UV technology allows for rapid fluorescence detection, providing an unmatched measurement speed and quality. And this therefore yields ultra-high resolution unfolding curves, which enable the detection of small, minute signals in the unfolding curve. So since there's no secondary reporter fluorophore required, unlike conventional DSF, protein solutions can be analyzed independent of buffer compositions and over a very wide concentration range, from samples exceeding 200 mg per mil down to 5 micrograms per milliliter. And this allows for the analysis of detergent solubilized membrane proteins, as well as highly concentrated antibody or enzyme formulations. With the Prometheus Panta, we also can assess the colloidal stability by observing the presence of large protein aggregates using back reflection technology to measure sample turbidity. So back reflection is going to measure the amount of light attenuated by a sample. And in the figure on the left, we have the cross-section of 2 capillaries that sit on the mirrored surface in the instrument. In a sample where there's no aggregation occurring, that incident light is reflected or directed into the sample capillary, and roughly 100% of that light is reflected back to the detector. In a sample where there is aggregation occurring, that incident light is reflected off those particles in solution so that not all of that light is reflected back to the detector, and it's effectively attenuated. And the amount of this attenuated light is then plotted as a function of temperature, shown here by the figure on the right, and therefore serves as a measure of the total aggregation in a sample. To probe oligomerization or small particles in solution, we're using the DLS optics in the Panta. So here, laser light is directed into the sample capillary and backscattered by particles in solution. The backscattered light is going to fluctuate in intensity due to the Brownian motion of the particles, and these fluctuations are measured and analyzed as a function of time. The raw data, or the autocorrelation function, describes the rate of these intensity fluctuations and determines the probability that a particle will be found in the same location over time tau. As larger particles undergo slower Brownian motion, they're going to have a slower autocorrelation decay, while faster, small moving particles are going to decay more rapidly. And by fitting this autocorrelation function, we can determine the diffusion coefficient, and then from the Stokes-Einstein relationship, we can calculate the hydrodynamic radius, or the particle size. So while the autocorrelation function tells us about the size of the particles in our sample, it also gives us the polydispersity index, or PDI, which is a measure of the sample heterogeneity of these particle sizes and can be used as a metric for sample quality. So in the figure on the top here, we have the cross-section of a capillary holding a monodispersed sample. And in the intensity distribution plot, we see one single very narrow sharp peak, which is indicating that there's only one particle size species present, and it has a very homogeneous, it's very monodisperse. Whereas the figure on the bottom, we have a polydispersed sample, and in the intensity distribution plot, we have two peaks indicating that there are two different particle species present, and these peaks are relatively broad, indicating that there's more sample heterogeneity for each of these particle species. To acquire a fluorescent sample signal in the Panta, capillaries are used as the sample containers, and each one holds 10 microliters of sample. These capillaries are going to sit directly on a thermal element, which is used to steadily heat the samples up to the desired end temperature according to a specified thermal gradient. And this direct contact of the capillaries with the thermal element results in more precise temperature control during measurements, so it helps to achieve more and higher reproducibility, not only between the capillaries, but also between different runs. So as the capillaries are heated, this entire tray is going to move beneath the optics that are fixed in the back of the instrument to constantly scan and acquire data points, which allows for the simultaneous measurements of both protein fluorescence, so thermal stability, aggregation, and DLS in a single thermal ring. And there are two optical heads in the Panta. The first one holds the fluorescence and back reflection LEDs in their respective detectors, and then the second optical head holds the DLS laser in its detector. So now this brings us to the second star of the show, the Monolith. And Monolith measures binding interactions using microscale thermophoresis, or MST. So microscale thermophoresis is a biophysical technique that allows you to measure the strength of the interaction between two molecules. And it does this by measuring changes in the fluorescence as a result of an IR laser-induced temperature change. And there are two components to an MST signal, and they are trick and thermophoresis. So trick, or temperature-related intensity change, is inherent to the fluorophore itself, where the change in the temperature is going to induce a change in the fluorescence intensity. And the fluorescence intensity is extremely sensitive to the chemical environment of the fluorophore. So whether or not a ligand is bound is going to affect the fluorophore's chemical environment, and therefore the extent of this fluorescence intensity change. Thermophoresis is the phenomenon where a biological molecule's movement in solution is directed by a temperature gradient. And the change in direction will affect the local concentration of the fluorophore and therefore the observed fluorescence. And the extent of this concentration change depends on a molecule's overall properties like its size, charge, and conformation. So there are two binding mechanisms that are going to affect the fluorophore's chemical environment and induce a change in the fluorescence intensity. And the first is through ligand proximity. So whether it's a small molecule, a peptide, or a protein, if the binding site is in close proximity to where the target has been fluorescently labeled, then binding will directly influence the local environment of the fluorophore. However, ligand binding often induces a conformational change in the target molecule. So even if the fluorophore has been labeled and is relatively distant from the ligand binding site on the target, since the fluorophore is incredibly sensitive to even subtle changes in its environment, this conformational change is going to affect how that fluorophore behaves and responds to a change in the temperature. In MST measurements, we are monitoring the fluorescence of the labeled target molecule that has been mixed with a dilution series of the ligand before and during the temperature increase. To induce MST, we modulate the temperature in solution with an infrared laser, and this laser is going to induce a very precise and reproducible temperature change for every titration point measured. Heating with this infrared laser is very fast, so we avoid disturbing the chemical equilibrium of the sample and saving a substantial amount of time. Initially, we are going to monitor the fluorescence of the sample while the IR laser is turned off to generate a baseline signal. And after a few seconds, this IR laser is going to be turned on, and you can start to see how the fluorescence changes over time. And the difference in the fluorescence detected between the bound and the unbound states defines this MST signal amplitude. So from these MST traces, we can extract some data and build a binding curve. And a typical MST experiment measures one MST trace for each capillary containing different concentrations of the ligand so that we can see how that ligand binding is affecting the signal. F0 here is going to be the fluorescence prior to turning the IR laser on, and F1 is going to be the IR laser on for a certain amount of time. F1 is then divided by F0 for each capillary's MST trace and is plotted as a function of the ligand concentration. And we can then fit this dose response curve here that you see populated on the right and extract a KD value from this fit. So today's e-demo, we will be highlighting the symbiotic relationship between the Prometheus Panta and the Monolith to probe the interaction between the receptor binding domain of the spike glycoprotein and the ACE2 receptor. So I'm sure you are all very familiar at this point with diagrams of the spike glycoprotein on the surface of the coronavirus. Entry of the coronavirus into the host cells is facilitated by the binding of the protein to an identified target, and in particular, today it's the cellular receptor ACE2. So since our MST experiment requires one of the binding partners to be labeled, the first step in our experimental workflow today is going to be determining the stability of the labeled target using the Panta's nano-DSF and DLS optics. An initial binding check is done on the Monolith. However, to further optimize our MST assay, a buffer screen is going to be performed on the Panta to identify the optimal buffer formulation and if any additives improve sample stability. And then finally, the binding assay is going to be performed on the Monolith to calculate an affinity. And with that, I will let my colleague Daniel Turman take it away.
Hi everyone, welcome to Nanotemper Laboratories in Cambridge, Massachusetts. I'm excited to be with you today to show you how our instruments interplay and allow you to be efficient as a scientist when you are interested in setting up assays for bonding experiments between proteins or protein small molecules or protein RNA, DNA, etc. Today we're going to be working with a sample that Leah introduced you to, the spike SARS-CoV-2 protein interacting with the ACE2 receptor. And to perform microscale thermophoresis, we need to label one of these, and we labeled the spike protein. That's going to be our target. And we're going to begin, I know that Leah mentioned that we will begin with the Panta. We're actually going to begin with the Monolith. We're going to perform an MST experiment. We're going to optimize using the Prometheus Panta. And then we're going to go back to the Monolith to look at our optimized binding samples. The data will be primarily shown when we go through the software after we kind of walk through the instruments, how they're loaded. And I want to do that now. So let's begin with the Monolith. The Monolith is about a 2x3 instrument, has a computer connected to it and a very large touchscreen display. I'm going to open this, and this is going to reveal the sample loading tray. Now the sample loading tray is detachable. That makes it very easy. I've got 6 capillaries there. So basically, you take off this metal holder and you place your capillaries into the slots like that. So this is a very efficient way and a quick way to do your experiments. One capillary at a time. What's really great, and as with most things with Nanotemper, we try to engineer them in ways that make it easier for you. We've spaced the capillaries exactly the space of a 384 well plate well. So what I mean by that, if we take our tilting station and we have a 384 well plate, we can easily dip our Monolith sample holder into that. So you could take 24 samples or 24 capillaries, dip it into one row of a 384 well plate. That will allow you to perform 2 12-point KD measurements. I'm going to place this back in the instrument. Or you can screen 6 buffers in that case, if you were doing screening. I placed the sample holder back on the instrument. It's controlled by a magnet and doesn't allow you to place it incorrectly. And I'm going to close the drawer. And that is it. That's all you have to do with your interaction with the Monolith when setting up experiments. It's buffer independent in terms of you can load anything in the capillary that will go in the capillary and perform the experiment. We'll go through the data collected here in just a moment. So the Monolith complements the Prometheus Panta. So with the Prometheus Panta, we can measure nano-DSF, aggregation, and dynamic light scattering, which gives us an idea about small and large particles in solution when they aggregate, and the stability of the protein in the particular buffer system that you're using. What's nice about Nanotemper products is that if you can use one, you can step to another one and be able to use it very quickly. And I'll show you why that's the case. So with the touch screen here, the instrument presents the sample loading tray for capillaries. And just the same way, we can take that tray off. It looks like this. There are grooves to put individual capillaries on, but the nice thing about the Panta, you can also place a 24 capillary chip space to load from a 384 well plate, just the same. I'm just going to place this back on. It's also controlled by magnets. Doesn't allow me to put it in incorrectly. And I'm going to close the instrument. So this is a quick walkthrough of how our instruments work together functionally, how they're loaded. And from here, I would like to take you back to the data in the instrument software so that we can look at how we took our SARS-CoV-2 and ACE2 receptor interaction from the Monolith, found that we had some problems with the assay setting up the microscale thermophoresis, optimized it on the Prometheus Panta, and then we went back to the Monolith and showed really nicely how we can optimize an assay in a morning, basically, and then you're set up for high throughput screening of small molecules, for instance, in a competition assay that would break that interaction between SARS-CoV-2 spike and ACE2 receptor. So I hope you enjoyed the quick walkthrough, and I'll be right back with a look at the software for you. So thanks for your attention, and we'll see you in just a moment. Hi everyone, welcome back to Nanotemper Laboratories, where we will look at some software analysis of the data we collected on the Monolith and the Panta of the SARS-CoV-2 spike protein interacting with the ACE2 receptor. Today we're going to look at two software modules, the Monolith affinity analysis and the Prometheus Panta analysis software. So these two softwares are suites that allow you to look deeper into your data. For the Monolith, that means you can look at your MST assays in an intuitive way that's user-friendly and very sophisticated. You can compare past assays. You can look at your dose response, and we'll go through that in just a moment. In the Panta analysis software, we're going to look at thermal unfolding data and aggregation data and dynamic light scattering data, and how that's presented to you all at once from an experiment that takes around an hour to an hour and 30 minutes, depending on your ramp speed, your temperature ramp speed. So I think you will enjoy and appreciate how the data looks, how we initially did the experiments on the affinity analysis software, moved to Panta analysis. We initially did the experiments on the Monolith, and then did experiments on the Panta to optimize our experiments for the Monolith. So we basically are showing you an ecosystem where you can take in one morning, optimize an assay of microscale thermophoresis on the Monolith and complement that with stability analysis. So you can put stability and function together in a buffer independent way, unlike any other system out there. So let's get started. We're going to first look at the Monolith experiments. So here's a lot of data in front of you now, but let's pick through it, figure out what it is. So it's an initial experiment measuring the spike protein with the ACE2 receptor. It's a microscale thermophoresis experiment, and that means we must label the target protein. And in this case, we labeled the spike protein with a red fluorophore, and we're using our Pico Red device, or Pico Red Monolith. Some exciting new aspects of our new Monolith is that you can get this in an intrinsic fluorescence, and in addition, the Pico Red model. Just keep that in mind. So here we have the spike ACE2 receptor domain. Let's walk through what an MST experiment will give you. So you begin with a equilibrium fluorescence measurement in the capillary, and we turn on the IR laser that heats the sample just a little bit, and you get a decay in the fluorescence signal. You get thermophoretic movement and then response of the fluorophore to the temperature gradient. And just looking at these traces, I can tell you that this is a borderline bad to terrible microscale thermophoresis experiment, because each trace does not decay in the same orderly fashion. You see how this one decays and comes out? Looks like almost there's a bump there. That's very indicative of aggregation in the sample. There's one more look we can take before we talk about the dose response. And the raw data is within the analysis software. And we got a lot of opportunities here to look at the quality of our recordings. So the first I want to just mention a capillary shape recording. So this is a relative fluorescence look at every capillary. We have 16 capillaries in this experiment. And what we're doing is scanning across the capillary, seeing the edge, and then seeing the other edge of the capillary. And we get a Batman look here to the capillary scan, and that is indicative of adsorption on the capillary walls. So just like was mentioned before in the wonderful presentations, you have this propensity of the protein at very low concentrations to stick to things, to stick to themselves, to aggregate, to stick to the walls of the capillary. And just really quickly, the conditions here are a Heapes buffer at 25 millimolar and pH 7.4 and 150 millimolar sodium chloride. So a physiological pH, near physiological sodium chloride levels. And in these conditions, it looks like our target protein sticks to the capillaries and aggregates in the solution. So these are very bad things for microscale thermophoresis experiments. But what do we do now? So we could screen many things and find a condition empirically that would give us better traces in the MST traces and would give us better capillary scans, indicating that we've removed the aggregation and the absorption. It turns out an even quicker way to go about this, and a more analytical way, is to use the Prometheus Panta to measure simultaneous nano-DSF to get colloidal stability, conformational stability, big and small aggregation information, big and small particles, and give us some idea about the stability of the protein, so we can come back to the monolith and measure the function of the binding experiments. So, quick note before we go there, from this data, we derive 1 point per curve. We do that by dividing this red zone, the value, average value, divided by the average value of the blue zone. And that gives us a F normalized value. So this ratio times 1,000 gives us 1 point, and these are different doses. We can measure this at any point, and we can measure this red window at any point in the MST trace. We're measuring at 5 seconds here because the software has an algorithm that optimizes when we should be doing this particular type of measurement. Here, the aggregation and the absorption is reflecting in the spread of the data, where you have a very large confidence interval, large standard error, and low signal to noise. So this is quite low. But we can fit it with a KD model. This is a KD model that's derived from the quadratic equation and has one free parameter, the KD, or the affinity. All right, so let's go now, since we got some nuts and bolts about this experiment, to the Panta and see if we can optimize our assay conditions to give us better data. So we're moving from an MST assay to a Prometheus Panta thermal unfolding assay. So the data looks a little different, and let's walk through it quickly so we can know what we're looking at, and then we can do some comparisons and learn some stuff about our protein. First of all, this is a technique that measures the intrinsic fluorescence of tryptophan. You don't need any labels. They can be there in the red region, and we're going to measure that in a moment, and it doesn't affect the tryptophan signal. It's a good reason why the ecosystem of the pico-red monolith works well with the Prometheus Panta. And we'll talk more about that in a moment. So this ratio measurement, 350 over 330, is the emission of tryptophan that's excited at 280. This gives us a nice, clean, unfolding sigmoidal curve that we can derive at the inflection point, the Tm. If you take the first derivative of this curve, you get a beautiful peak right at the inflection point, and that's what you're looking at here. Here's the turbidity optics. This gives you large-scale aggregation. Basically, it's a measure of colloidal stability. And in this particular sample, there is no breakdown completely of the solution where you can see a large-scale turbid response. However, if you look at the dynamic light scattering, the cumulant radius analysis is very telling. So this is a look at the hydrodynamic radius of the protein. It's quite small protein, starts at around 3, a little less than 3 nanometers. And as we go along, we have a marker here. That's the onset. That's when the size begins to change. It's at a quite low temperature. And you get twofold log changes out here in the 60 to 70, but those changes are small here, but there are changes. So you begin to see some aggregation of the protein. So there is some colloidal stability issues in HEPEs and sodium chloride here. That's what B1 is in our buffer scouting experiment that we're doing today. So let's think back about what the presenters told us earlier. So our colloidal stability and conformational stability measurements here indicate that we have some adsorption on the capillary from the Monolith data, and we have some aggregation that's showing up at low temperatures on the Prometheus. I think that a good way to get around this is to add something that excludes the protein, like a surfactant or like glycerol. So let's try that. Buffer 3 is an addition, or B3 is an addition of glycerol. B2 is the addition of a surfactant, pluronic F127. And B4 is addition of both. But before we get there, we did a maneuver with our protein to measure it in the monolith. And that maneuver was labeling the lysines with an NHS red dye. Almost always we get the question of, does labeling your protein affect the stability, therefore affecting the function? This is a wonderful way to answer that question. If it affects the stability, we would expect the inflection point to shift to the left to a lower temperature. The conformational stability, if it affects the colloidal stability, it should aggregate at a lower temperature. So let's compare, before we get into the buffers, the labeled protein with the native protein. And what we see is really no differences in the conformational or colloidal stability. So this is great news. We have a protein that we have attached a label on the lysines. And this allows us to say definitively we have not affected the stability of the protein, therefore we haven't affected the function. So let's take this away and let's have a look at the addition of glycerol. This is 10% glycerol. And now we're looking for changes in stability. And what we see is we have nicely changed the colloidal stability properties of the solution, but we have made no changes to the conformational stability. What happens if we add in B2 a surfactant? Now this is quite beautiful. We get a nice conformational stability improvement here, but we still have the colloidal stability problem. So let's take the addition of glycerol and a surfactant and see if we fix both of these problems. And in fact, we do. We still get the improvement of conformational stability and the improvement of colloidal stability. So the indications here is that we should move on in our assays with a buffer that includes glycerol and a surfactant, chloronic F127, and take these conditions back to the monolith and see if it optimizes our assay. So this experiment took an hour and 10 minutes, very quick. And we have beautiful confirmatory data that our functionality isn't affected by labeling with NHS dye at lysines. And we have a buffer that's going to give us a better monolith MST trace. So let's go back to the affinity analysis software and have a look. So our initial recordings were aggregation, indications of aggregation, and we also had Batman-looking capillary scans. So let's look at our optimized B4 buffer with glycerol and chloronic, and this is quite incredible. We get very, very beautiful MST traces. We've completely taken away the aggregation. And then when we look at the raw data, we get very nice traces. We no longer have the Batman peaks. And so I'm very happy with this. We've taken, over the span of 2 to 3 hours, a critical interaction to our society today. The spike SARS-CoV-2 protein interacting with the ACE2 receptor. Optimize the assay. And now if we wanted, we could move forward and watching and trying to introduce drugs to see if we break up that interaction. But before I get ahead of myself, let's have a closer look at the data. So the capillary scans look great. The dose response looks far better than before. So our response amplitude is beautiful. We have a much tighter confidence interval. Standard error is 3-fold lower. Signal to noise is 3-fold higher. We have a KD of 1 nanomolar. This is spot on what the KD is of this interaction measured by ITC, SPR, FP. Many different techniques have been used to measure this interaction at this point. So I hope you enjoyed our look through the ecosystem of NanoTemper's instruments, the Monolith and the Panta. And if you're interested in measuring the stability and the function of your proteins and their interactions with ligands, I think this ecosystem of the Monolith and the Panta that NanoTemper has gives you a small footprint and a bigger bang and allows you to do this quickly. So you can optimize your assays and move on with your important science. So thank you.